Okay, so welcome to my notebook. We're just gonna go back to the front page and a more formal welcome will be here shortly. Alright. Okay, so hi everyone, or just one person maybe. This is, or I hope this is, a brief interview of the cell cycle and its relation to cancer. In this video, we'll go over the phases of the cell cycle, um, what controls its processes, as well as what happens when those processes are disrupted. So, looking at this blue area right here, that is interphase, where this cell spends most of its life. The G1 phase is when the cell is maturing. The S phase is where the DNA is replicating, and the G2 phase is when the cell is further preparing for mitosis. An example would be making microtubules to pull the chromosomes apart in mitosis, which is kind of what's happening right here. Also, a little extra phase is the G0 phase, which is more of a resting stage, maybe, if the cell hasn't quite made it through, or an eternal state where a cell is not dividing, nor is that particular cell preparing to divide. And those examples could be a neuron or a very sad attempt at a striated muscle. So once we reach cell division, I'd like to make a little note off to the side that binary fission is only for prokaryotes. When we talk about cell division right now, we're talking about it in the case for eukaryotes. So the end goal of mitosis is to pass a complete set of genomes from the parent to the daughter cells. The replicated chromosomes are separated into two nuclei. Note that these are spindle fibers that move the chromosomes to opposite poles of the cell. And right here for cytokinesis, looking at this cleavage spur over there, that is the physical division of the cytoplasm of the parent cell into two identical daughter cells. And then we get to see later right here, the checkpoints, which are the regulators of the cell cycle. So, just looking at this circle right here, we see the G1, the G2, and the spindle checkpoints. The G1 checkpoint is also known as the cell growth checkpoint. And some important things to note, though it's not always the most important, it just depends on the type of cell, however, but this checkpoint would look for such things as whether or not the cell is large enough, whether or not the cell has enough nutrients or energy to power through. If the cell is receiving messages from neighbor cells, probably in the form of growth factors, or if the DNA of the cell is damaged. If the cell doesn't get the pass, it enters the G0 phase, made apparent by this purple arrow right here, which again is a resting stage, or for some cells, the perennial state. The G2 checkpoint is the DNA synthesis checkpoint. The checkpoints make sure that the daughter cells will receive healthy DNA, so this checkpoint will make sure that the integrity of the DNA is adequate and the DNA has replicated properly. The spindle checkpoint, or the mitosis checkpoint, or the M phase checkpoint, checks to see that the spindle fibers are securely attached to the sister chromatids. And this cycle will not continue until the chromosomes have attachments from two spindle fibers at opposite poles of the cell. And there are also internal and external signals that help regulate the process as well. So we'll zoom in a little bit to internal signals, though it's kind of small, it's not as important. in here, but it is. Okay, so an example of external signals are protein signals, which helps the cells know when and when not to divide. An example of this is platelet-derived growth factors, or PDGFs, which is made by platelets in blood clots. PDGFs will communicate with nearby cells to stimulate cell division and to heal wounds. And then we're just going to move on here to internal signals. An example of internal signals will be cyclins and cyclin-dependent kinases, or right here, if I can just find my mouse, CDKs. So, looking at this, for now, at least in this area, when a cyclin binds to a CDK, it activates it, making the kinase a functional enzyme, and it allows the kinase to modify target proteins by attaching phosphate groups to that target. Oh shoot, that's supposed to be a 3. But that's important. But what is important is to note that CDK levels stay constant throughout the cycle. The levels of activity and protein fluctuate with the levels of cyclin, seen here by this graph. And even specific cyclins are made at specific phases of the cell, or highs at specific phases of the cell. And a great example of cyclin at work is the M cyclin, the mitosis promoting factor. This is responsible for moving the cell past the G2 checkpoint into the M phase. The M cyclin, as seen right here, would build up, and when it reaches the point where the G2 phase transitions to the M phase, it will bind with the CDK to form a complex. The complex adds phosphates to the different proteins in the nuclear envelope, which is seen by this diagram right here, to the different proteins. And that is important for the beginning stages of the M phase, or to other target or the complex itself, will add phosphates to other target proteins, which can create an action that calls for M phase events. Oh goodness. And when these processes seem to find a disruption of some sort, it leads to something called cancer. And cancer, quite simply, is abnormal cell division or cell growth. These cells can multiply in a culture, for example, without the need for growth factors, because they make their own growth factors, or their growth factors are constantly on. Oh my goodness. Or the receptors are constantly on, or neighboring cells are tricked into producing factors for them. A normal cell, or normal versions of cellular genes, are called proto-oncogenes. And they code for proteins that simulate normal cell growth and division. However, environmental cancer-causing agents, or just cancer-causing agents in general, can damage a normal cell and cause it to be a cancer cell by mutating that specific gene, turning it into an oncogene. And several examples would be the RAS protein and the p53. So, starting with the RAS protein, or the RAS protein, I don't know why I say RAS. It is a G protein, and a normal pathway is that it makes a final response for a protein that simulates the cell cycle made clear by the cell division and proliferation. In the event that there is a mutation in which the RAS protein becomes hyperactive, um, it it triggers the transduction cascade um, without growth factors. Right here, without growth factors. There is an overexpression of the final protein because the mutant version of the protein is always on. So there will be more cell growth, more abnormal cell growth. Yet another example is the p53 protein. That needs to stop, sorry. Is the p53 protein. It is a tumor suppressor, and I saw this earlier, but it's also known as the guardian of the genome. In the event that DNA is damaged, it will stop the cycle at the G1 checkpoint by triggering the CDK inhibitor to block the CDK and cyclin complex. And that would stop the cycle from progressing. And by doing that, it gives it time for the actual p53 protein to repair the DNA. In the event that the DNA cannot be repaired, it will trigger apoptosis, and the cell doesn't pass on the damaged DNA. But in the event that the p53 protein is non-functional or defective, it allows for the mutations to happen rather quickly, which can ultimately lead to cancer. And I think that concludes this little YouTube video. I hope it involves some helpful stuff, and I hope that you have a good day or a good